Our next speaker is Nguyen, who is going to talk about a novel antigen detection assay for diagnosis of and use in advanced HIV disease in Vietnam. She is a senior research associate in infectious diseases. Welcome.
Okay, thank you. Good morning. I'm so excited to sharing our exciting and new data about a novel antigen assay for diagnosis Talaromycosis. So Talaromyces, formerly named Penicillium malefii, is one of the 7 dimorphic fungal pathogens causing the major global mortality and morbidity. It causes invasive fungal infection involving liver, spleen, bone marrow, and bloodstream. It is a leading cause of HIV-associated death in Vietnam and southern China, with annual incidence of 8,000 cases. And late diagnosis is still the more challenging clinical problem. Why? Because the current gold standard diagnosis is still on histopathology, which is invasive but not frequently done, and also on culture, mostly from blood, and the sensitivity is 70%, and the positive result come to the patient often at a late stage. The molecular diagnosis is faster, but the sensitivity as high as the blood culture is meaning 70%. Okay, thank you. But this is require the sophisticated machine, and lately antigen detection is now the standard of care of cryptococcosis and aspergillosis, and it is present a promising diagnostic model for all fungal infection because it's simple and accuracy. About the antigen detection, the keyword is about the discovery of the one protein is unique for this pathogen. This was found by a group in Hong Kong University, and this protein is MP1. It belongs to the glycomannoprotein group with this structure here, and it's expressed a lot on the cell wall, and it is released abundantly in the bloodstream, which is ideal for the serological diagnosis. This protein is also immunogenic, which means that they have the binding site with the arachidonic acid, the key element for pro-inflammatory intermediate. And also from the mouse model, they also show that this protein is also the virulence factor. So for this antigen, so we collaborated with the Hong Kong University and developed the enzyme immunoassay on our cohort from 372 culture-confirmed Talaromycosis and 517 non-Talaromycosis control. The ROC plotted from the true positive and against false negative, and the cutoff is at 0.5 determined by Uden index, which is maximize the true positive and minimize the false positive, which gives the sensitivity is 86%, which is higher than blood culture is 70%, and excellent specificity is 98%. But so what about like this antigen? This assay can detect the antigenemia in the asymptomatic patient. So we evaluate, we use the assay screen from over 8,000 asymptomatic patients in 2 provinces in the southern of China, and the TM antigen prevalence is 9.4%. And in Vietnam, screening from over 1,000 asymptomatic patients with a CD4 less than 100, and the TM antigen prevalence is 4.2%. But the clinical information from these patients are not available, so we don't know that the TM antigenemia means clinically. So with this information lead the research hypothesis. So from the infection initiated by inhalation of spore, and from the infection, the patient can develop dissemination infection for years, or for many years, depending on the immune system. So the patient can be either asymptomatic or non-specific symptoms. And from this dissemination, patient can develop the late clinical manifestation, which is at which time the culture will be positive. So we hypothesize that based on our retrospective data, so we hypothesize that using this assay, this assay will be more sensitive than culture and reduce substantially the time to diagnosis. And then additionally, from the TM antigen screening data, we think that using this assay, we can detect the patient that have antigenemia that are at risk at developing disease. And then using assay at this dissemination, we can detect the patient at the early of infection and can give the preventive treatment before the disease can develop. So to test the hypothesis, we recruit all hospitalized patients at the age over 18 with CD4 less than 100 with any symptoms, and for having a power to differentiate, to detect a different of sensitivity of 20%, so the sample size will be 520. So at the enrollment, the serum, plasma, and urine is collected to run the EIA and follow twice monthly for developing disease over 6 months. Alongside with the ELISA, the routine culture from blood and other bodily fluids were performed at enrollment and as clinically indicated during the follow-up. The primary outcome are diagnostic values, include sensitivity, specificity, positive predictive value, negative predictive value. Based on the gold standard reference, it is defined as the incidence of culture-confirmed TM from any body fluids like blood, skin lesion, bone marrow for over 6 months. So from 1.5 years, from January 2018 to July 2019, there were 533 patients were recruited, and 452 patients didn't develop the disease over the 6 months. There were 16 patients who have false positive, which have the TM antigen positive but didn't develop disease. So which give the specificity is 96.5%. So there were like 81 patients who have the culture positive disease. So of those, 96 patients have culture positive during the enrollment, which give the sensitivity of culture method is 85%. So 12 patients have developed disease over the 6 months, and all of them had TM antigen positive. So in those patients who have culture positive during the enrollment, the assay only missed 2 patients, which give the overall sensitivity is 97.5%, which is higher than culture method, and the difference is statistically significant. So for those patients who have a TM antigen positive but the culture negative, so the time to have the culture positive range from 1 week to 16 weeks, and the majority is from week 3 and week 4. So this assay can be detected the infection up to 16 weeks before the cultures become positive. So we want to know that which clinical feature is associated with the having positive TM antigen test. So we evaluate 9 variables, and 7 out of 9 variables are independent predictor for the TM positive. So they are the residency or travel in the Central Highland, presenting skin lesion, have CD4 count less than 50, low lymphocyte less than 500, low platelet less than 550, and elevate AST. So this will help us to narrow down the number of patients at risk for screening in the future and reduce the cost. So which is the best specimen to diagnose the infection? So this table shows like the 2 negative, 2 positive, 4 positive, and 4 negative and 4 positive between 3 specimens. So the number of false negative is 8 in serum, 7 in plasma, but only 4 in urine. And the same, similarly, in the number of false positive, the urine also have the lowest false positive, which give the urine as a high sensitivity and high specificity. The OD distribution, the graph below also demonstrate that urine is the best metric for differentiate the positive and negative. So there were 480 patients whom we all have sera, plasma, and urine from the same patient. So we want to compare the diagnostic performance between these specimens, and we found that the accuracy between 3 specimens is the same, is similar and very high, about 95%. For the sensitivity and the specificity is very high in urine, with the sensitivity is 94.4 for urine and for sensitivity, and 98.8 specificity for urine, and it is higher than plasma and sera. But the difference is statistically different with the specificity. So in the conclusion, we think that this assay can detect infection up to 16 weeks before the culture becomes positive. And this assay can be more sensitive than culture, 98% versus 85%, and with a very high specificity. And then urine is superior to plasma and sera in both sensitivity and specificity. For the future direction, we want to build the evidence to bring the antigen test to the bedside, and we evaluate the TM antigen test as a screening tool for asymptomatic infection. And we build the evidence for a screen and treat paradigm. And finally, we want to collaborate with the industry to develop the commercial point-of-care test. And finally, I wanted to say the thank to all the patients to participate to this study, the investigator in Vietnam, all our collaborator in Hong Kong University, and at Duke. I want to say the thank to my supervisor, Dr. Thuyle and Dr. John Perfect. And finally, thank you for CFAR and NIH and all the funder. And finally, thank you for your attention.
Really exciting, thank you. Can you comment, do you still think you need a confirmatory blood test as well, a culture test?
Yeah, so you mean that the confirm plus test for all the fungal infections, right?
Yeah, would you
Yes.
still do a culture to confirm?
That's right. So right now, the culture to eliminate or like to remove all the fungal infection is not easy. So we try the best to collect all of the clinical information as well as like the blood culture and then the symptom to know that patient doesn't have all the fungal infection. That is the best that we can do right now. Yes.
Any other questions? Thank you very much.
Yes, thank you.
So before I turn it over to Kent, who's going to, I guess, introduce the poster session, I just want to see if Maria Blasi is here. We missed you a little bit earlier. Somebody started walking. Thank you. You won the award as well, so we want to make sure you got the plaque and had the opportunity for a picture. Thank you for all your work.
Thank you.